So I'm Viola Vogel. I am professor at ETH Zürich, heading a Laboratory of Applied Mechanobiology. So we are totally interested how mechanical forces change the function of proteins and how that impacts cell signaling, but then also how tissues are forming, how tissues are repaired, and how the tension of tissue fibers is different in health and disease. So we have a number of favorite molecules. Favorite molecules include the molecules involved in fertilization that form the very, very first specific contact between a sperm and an egg. And there we are very excited to show that mechanical forces are necessary to create a firm attachment of a sperm. And the beginning of life does not happen in equilibrium. Instead, the sperm is really propelled by the flagellum, and with that, a lot of forces are applied, tensile forces are applied to the first contact, and we discovered recently that the first proteins, Juno and Isomo, form a catch bond. No, it's the latest. We discovered that catch bonds exist many, many years ago, nearly 25 years ago, between bacterial adhesins and sugar mannose-coated surfaces or mannoses on cell surfaces. And afterwards, a lot of other catch bonds have been identified, for example, formed by integrins when they adhere to extracellular matrix. So catch bonds allow a lifetime extension if forces are applied to a receptor-ligand interaction. Typical slip bonds, the receptor just separated more easily under force from the ligand. However, catch bonds have an extended lifetime, which also allows cells to adhere to surfaces or via caterions to pull on each other. So one central technology that helped us to develop a lot of structural insights is steered molecular dynamics simulation. And these simulations are necessary because all knowledge we have about proteins is essentially equilibrium knowledge of their structures. And if we want to understand how the functions are switched by stretching proteins, we need to have access to mechanically stabilized intermediate states that might play by many not so appreciated function in life. We work since a long time with fibronectin, and that's certainly our pet molecule. And we never thought that we would work on that for decades. But fibronectin is a huge glycoproteins with many, many domains. And each domain is furbished with another binding site for binding partners. And those can be cells, they can be bacteria, and growth factors, and many other proteins that play central roles in regulating cell functions. And so we mapped out that many of these binding sites are indeed force-regulated. I was very lucky. I had many mentors. From my first steps deciding what I wanted to study, and I picked physics and biology, which was not very common at that stage. And so there, a friend of my parents was a physicist and had a huge impact on me wanting to study physics. But then as I started my career in bioengineering at the University of Washington in Seattle, Buddy Ratner, working on biomaterials, was a very, very good mentor to me. And then Frances Arnold, whom I met earlier at the conference, we are very close friends since then, because there were so few women in engineering that pursued academic careers at that time, and many more in my life. So one central question, and that was raised often now at the meeting, is once the extracellular matrix gets degraded, once it gets remodeled during disease, we very often focus on how to change cell signaling or cell fate, but not so often about how we can repair the extracellular matrix in very efficient ways. And so I think the core strength of this community, the extracellular matrix community, is to raise attention to the fact that matrix biology should not be neglected and plays a major role in guiding cell function. So it's the reciprocal interaction between cells and their matrix, which they make and transform, that finally decides whether we stay healthy or get sick with one or another progressive disease. I think what is crucial is to always look for new techniques, to not be blinded by the type of training that we received, to look out for other knowledge from other fields. I think a lot has been done by creating a lot of interdisciplinary study programs, but it's a lifelong learning process. And particularly now we see again how our entire world is transforming with the advent of AI and all the new possibilities. So to always think very carefully, how can we combine new technology and apply the new technologies to learn something that was previously completely inaccessible. So what is very nice to see in this meeting is that many of the researchers involved have started companies and still are sharing very openly where they stand. And extracellular matrix is so complex, involving so many molecules. And we very often approach a question from the insights from one molecule or one signaling pathway. And the complementarity of all the knowledge that is also presented here again. And the free exchange is crucial for us to make progress and not to oversee a metabolic pathway that is important. Similarly to mechanoregulation, similarly to the different classes of extracellular matrix proteins and how they are related to diseases, and how much can be learned from point mutations in humans that allows us to better understand how structural mechanisms that we find for humans are different from mouse models. And for us, or for me, working in bioengineering, that is certainly very important, that people do not stop after they show the mechanism in a mouse, but continue bringing it into the clinic. I feel that at a time where so many people have dogs at home and cats, and it would be nice if scientists could learn more from all the pets that are around. For example, many pets, once they get sick, people do not try to treat them, or it's too expensive. Why not try some of the treatments experimentally if people volunteer that their pets are being treated? I think that would save a lot of money and would give us insights that we can't just get from mouse experiments.